All right, so I forgot to record for a second. We started with our replication. Our goal of replication is to make a second copy of DNA for mitosis. DNA never ever leaves the nucleus, so to make a second copy of it, we need to be in the nucleus. And to do this, we have two players. The helicase is going to unzip our DNA, and then DNA polymerase is going to come and connect my nucleotides, my A's to my T's, my G's to my C's. If we follow the order of my players, it tells me the steps in replication. So DNA helicase unzips. So stage 1 was to unzip my DNA. And then my DNA polymerase came along and connected my nucleotides. So stage 2 was to pair up and build second strand. So we unzipped our DNA, and then DNA polymerase came and would pair my A's to my T's, my G's to my C's, and connect and turn one strand into two. So what was my end product? What did I end up with? My goal was to make more DNA, so I ended up with more DNA. How many DNA strands did I end up with? One more, so 2 total. So now I have 2 identical DNA strands. So that is my end result, is to have 2 identical DNA strands. So that is replication, which entirely happens in the S phase of interphase of a cell's life in preparation for mitosis. All right, so then now let's look at my 2 stages of protein synthesis. When we say central dogma, we're saying basically the common practice, the common theory for molecular biology or for biology. And that is the idea that our DNA makes mRNA, our mRNA helps make our protein. So the main goal is DNA getting to our protein. So let's start with step 1, transcription. What is my goal in transcription? Go from DNA to, beautiful. It's right there for you. DNA. So we're going to transcribe DNA into mRNA. It's fine. Transcribe is just a fancy way of saying I'm going to take DNA's message and I'm going to put it into mRNA. If you ever go to court, there's always a transcriber. That's that person that's sitting there pushing all those buttons really fast and it has a weird looking keyboard. That's them transcribing the conversation that's going on in the court. All right, so I have DNA. So what organelle must I be in? The nucleus, because DNA never, ever, ever leaves the nucleus. But my mRNA will, which is why I'm making my mRNA. You can guess. It is. So I'm not pulling you into the enzymes. I'm not making you do the enzymes for this one, but you have your DNA because we need to make a copy of it. And then we have our mRNA, which is going to take our message outside of the cell. There aren't too many steps to this one. Okay. Not quite. We're getting there, but we're not quite to proteins. No. All right, so my first step, I'm going to have to unzip my DNA again because I need to get in between the strands to be able to make my message. And now I'm going to make my mRNA by pairing nucleotides with the DNA. But no, which nucleotide do I not have in mRNA? The T. There's no thymine, no T's. What do I have instead? A U. I have uracil instead of thymine. So replication and transcription are actually very similar in what they're doing. It's just one is making a second copy of DNA. The other one is making an mRNA, which has a very similar or has the exact same message as the DNA. And then my final step here, my mRNA will leave the nucleus. So DNA does not leave, but my mRNA, my messenger RNA, is going to go take the message. You can think of this a lot like a king in his castle. The king knows all. He is the one that directs everyone and tells them what to do, but he very rarely leaves the castle. However, someone needs to take the message to the people, and that is where messenger RNA comes in. He comes to the king, he hears his message, he gets on his horse, he leaves the castle. The king is safe and protected. Doesn't matter if the dude on the horse dies because he's not the king. I mean, we still care about him, but not as important as the king. So messenger RNA rides off on his horse to tell the people what to do. So what is my product? What did I make? Who is my horse riding off? Messenger. So my end result is mRNA. My end is my mRNA. Have I made a protein? Have I? No, I've only made mRNA. I have my message. My message is going out into the world, but someone needs to hear the message. So on to translation. What is my goal of translation? Is to make protein. My goal is to make a protein. Am I in the nucleus still? No, my horse has ridden off. He's left the castle. He is heading to the people. Where is my messenger RNA going to go to make a protein? Not quite. It's in the cytoplasm. It's another organelle. Ribosome. We're heading off to our ribosome. My ribosome is my protein making factory. So my players. I obviously have a ribosome. You can kind of think of him like a typewriter. He kind of moves the message along, making sure everything lines up properly. I have my messenger RNA. He's bringing me my message, so I know how to make my protein. He's kind of my recipe. And then who brings me my ingredients? tRNA. My tRNA transfer RNA is going to transfer amino acids to make my protein. So my tRNA is actually connected to an amino acid, which we'll see here in a little bit how that connects. And you'll see it a lot more tomorrow when we talk about codons. Amino acid makes a protein. It's the monomer of a protein. So tons of amino acids connected. One single amino acid is not a protein. Amino acids connected are a protein. If I just have a bowl of amino acids that aren't connected, they are not a protein. They must be bonded together. I imagine them just bubbling around. It's kind of like right now, there's hydrogen in the air and there's oxygen in the air, but it's not raining. When they combine, they'll make water. But until they're bonded, they are not water. So amino acids can bubble. Like right now, your body is just filled with amino acids. But it's not until your body calls upon them to bind together that they become proteins. Does that make sense? Okay. All right. My steps. So my mRNA will be connected in the ribosome. So my mRNA goes and it finds my ribosome. We're going to see today ribosome's going to kind of clamp onto it. When my mRNA is clamped into my ribosome, it kind of sends out a message to my tRNA. And it's going to start reading those 3 letters, those codons. And so what happens then are tRNA that match the codon come to the ribosome. So let's say I have CCC. I'm going to be sending out a message saying, I'm looking for a tRNA that has GGG. I want them to be complementary, just like what we did in your review worksheet. So CCC, and then there's a little tRNA over here. He's got a GGG. He comes and he connects with the codon of the DNA, bringing that amino acid. And so then my amino acid on the tRNA bonds to start making proteins. So this is going to happen over and over and over again. tRNA brings in another amino acid, then another, and they're all bonding together, starting to look like a pearl necklace, beads on a string. It's going to do this until it reaches the stop codon, which we will see tomorrow. You saw a little bit of it in your DNA alias. You always tacked on tag to the end. That's your stop codon, one of your stop codons. Once we reach the stop codon, we release the protein. And all is good in the world. I was wondering what you were doing. We're going to see this here in a second, too. We're going to do kind of a together, make a sentence, hidden message thing. Don't you know? Yep. 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 So you just need the steps. All right. What is my product of translation? What did I make? A protein. That's my product. I have made a protein. So, my king gave my messenger a message. My message rode out to the townspeople and said, townspeople, we need more ditches. I don't know. And the townspeople said, okay. And they worked together. They made some ditches. That's your DNA, your protein synthesis. My king is my DNA. My horse person is my messenger, my mRNA. My message is given to them based on what I need. I needed ditches. We made a ditch. Now, imagine everything that you could possibly need for a kingdom. Houses, ditches, food, animal care, weapons, armies, doctors. Did my message have all of those? No, my message was just to make a ditch. My mRNA is going to be one, maybe a couple genes at a time. My mRNA is not my entire strand of DNA. It's only whatever I need at that moment. Otherwise, you would burn a lot of energy for no reason and you would always be exhausted. Questions about our central dogma graphic organizer and the things that you will be tested on very shortly, probably next Monday. Questions? I'm just going to turn off the recording.